In this video, also for exam 4, we're comparing 3 enzymes. We're going to compare DNA polymerase and ATP synthase and the ATPase domain of myosin. And we'll be looking at the chemical reaction, the chemistry. And for DNA polymerase, we're taking a primer DNA strand with its 3' hydroxyl group and combining that with an activated nucleotide triphosphate in the deoxy form, so a DNTP. And then we're creating a DNA primer, which is one longer, N+1, and has also a 3' hydroxyl group for the next round of nucleotidal transfer reaction. And the leaving group in this reaction is pyrophosphate, PPI. For ATP synthase, we're combining ADP plus inorganic phosphate, and we're creating a molecule of ATP. And the leaving group here is water. And for myosin, it's exactly the same reaction, but we'll write it out in reverse to emphasize that we're hydrolyzing ATP plus inorganic phosphate. And there's movement, some kind of moving parts in each case. And for myosin, movement is a type of linear walking movement along actin filaments. And the chemical potential energy represented by this reaction is driving the walking along actin. For ATP synthase, the movement is a circular motion. Circular motion of the gamma subunit, which drives these transitions LTO. And that drives the release of the ATP from the beta subunit in ATP synthase. And for DNA polymerase, there's also a movement. It's the movement of the O helix. And the purpose of this movement is to ensure accuracy or fidelity. Fidelity of the choice. The template directs which nucleotide we're accepting for this chemistry, and that choice is made more accurate by movement of the O helix. To try to illustrate what I mean by that, I'm going to use some puzzle pieces. Over here we have puzzle, Henri Matisse, purple robe, and anemones. And here we have a portion of the puzzle already fabricated, and we're going to take the puzzle piece apart so we have some parts to work with. And you can think of these 2 rows of puzzle pieces as the template strand and the primer strand of DNA. And if we're trying to select the next nucleotide triphosphate to come in here, we often are going to encounter mismatches that don't quite fit. And when I push my thumb down, that's the equivalent of the O helix pressing on the newly formed base pair. If we get the right piece, when the O helix presses down, it works and it can fit all the way into place. And that then allows the chemistry of forming this covalent bond between the 3' hydroxyl group and the alpha phosphate of the incoming nucleotide triphosphate. If we use the O helix movement by analogy with what we naturally do when we're building puzzles, we see that sometimes we get a pretty close fit, but it doesn't pop completely in place. And that is the equivalent of rejecting the wrong choice for the synthesis of DNA. And we spend a lot of time rejecting the wrong choice. DNA polymerase gets the right nucleotide 1 out of every 4 attempts. And so the O helix is prevented from closing over the nucleotide more often than it actually finishes the job and closes over.